Hi everyone, it's Mr. Sinti, and today I'd like to talk to you a little bit about meiosis and Sordaria. Sordaria is a particular fungus, and it's quite useful in studying inheritance. And so this video is about that. And so I want to say that at the top of the video, I want to talk a little bit about a little bit of general knowledge in fungi in case you're unfamiliar with it. And then I'm going to sort of transition into a discussion of meiosis as it leads to the spores of Sordaria, how they reproduce. And then I'm going to conclude with kind of a discussion of how those spores can lead us to looking at frequency of recombinance. In other words, how often does crossover occur, and therefore one could deduce a map unit in terms of the location of this particular gene that creates spore color in fungi. So hopefully that sounds intriguing. You'll stay tuned. So right out of the gate, fungi so important, and so ecosystems would be in big trouble without fungi. So as you know, they decompose dead organisms and leaves and break down materials, and they recycle all of these organic molecules into the most simple elements so that they can be then assimilated by plants in particular. And so they're very critical. I just want to point that out. And a lot of plants, speaking of plants, rely on fungi, especially in their root system, mutualistically, in order to increase the surface area of the roots so that they can gather more nutrients and more water from the soil. So that's really important for plants. Humans, we've always loved fungi, not only just for mushrooms, but we've cultivated them for centuries for food, antibiotics, as you can see here, destroying bacteria, the discovery of antibiotics, very important from fungi, and then of course yeast, making our bread rise and fermenting sugars for beer and wine, most important. And so I also wanted to say something about fungi in case you didn't know that they're eukaryotic organisms, and they're mostly multicellular. And so curiously, long ago they were classified in terms of taxonomy as being more related to plants. Though they don't photosynthesize, they seemed plant-like in some ways. In other words, they didn't eat per se. They derived their nutrients from digestion of extracellular digestion and decomposition. But in fact, more recently, when you look at DNA sequences, they're more closely related to animals than to plants. And I find that can be interesting. And so speaking of nutrition and absorption, fungi, they can live on any sort of substrate. Here that you can see these fungus growing on the side of a plant, a tree bark, or they can grow in the soil. But basically, they absorb their nutrition. They absorb it. And so they must then decompose or digest material using exoenzymes. In other words, they secrete these hydrolytic enzymes to the outside of their cells. They break down the food, and then they absorb them through diffusion. So they're heterotrophic. They don't make their own food. They get their food from other sources. And so how do they go about doing this? Well, if we look a little bit at the vegetative body of a fungus, they're composed of these little tiny microscopic filaments called hyphae. And again, an alert to when you're approaching something like fungi, there's going to be some unique terminology that is helpful, but don't be overwhelmed by that. So these hyphae are sort of like the primitive root system of a fungus, and so it helps to increase surface area. And then collectively they form this sort of interwoven mat called the mycelium. So all of this is how the nutrients are then absorbed from the soil right there, and this is below the ground. So the fungi can be huge. Okay, so we're talking about like they and you don't really notice this because this is subterranean below the ground. But let me just possibly shock you with this. So up in Oregon, okay, so right around this area in Oregon right here, there's a particular giant fungi that is, you ready for this? 3.4 miles in diameter, and it's covering over 2,000 acres. And so this thing is thousands of years old, and it's huge. It weighs hundreds of tons. So I always, if you know me, you know I'm a big fan of the coastal redwood, and I always like to I believe that it's the tallest and largest living thing. But the truth is, the fungi are pretty massive, and this might be the largest living thing ever on the earth. And so these fungal hyphae, here you can see under the microscope 1000x. This is what is secreting the enzymes, this is what's breaking down things, and this is how they're absorbing. Now, the walls, just to point out, because it's kind of interesting, the walls of the hyphae, or walls of the cell wall of a fungi, is made up of chitin. And if you're familiar with this, this chitin is it's kind of a flexible polysaccharide because it has some nitrogenous functional groups associated with it. It's the same material that's found in the animal kingdom in phylum arthropods. So things like crab, lobster, and insects have the same sort of exoskeleton made up of chitin. So that's kind of an interesting thing. Again, I was mentioning how the hyphae, as you can see here, form these really elaborate networks to increase surface area called mycelium that provides, again, surface area for absorption. Let's talk a little bit about reproduction, because the emphasis of the video is going to be on meiosis and the production of spores in fungi. And so they produce spores either sexually or asexually, and let's talk about that. And so these spores are the reproductive structure, and they're enormous in terms of numbers, reaching in the trillions. And so they produce a lot of these spores, which is indicative of the fact that they often are not necessarily successful. They're transmitted by wind and by water, and when they land on the soil, and if it's moist enough and they find enough food, then they'll start to grow and produce their own mycelium. Okay, and so I want to focus in, that was a very brief introduction to fungi. I want to focus in to a particular phylum within the fungi kingdom called Asciomycota. And what's characteristic about this particular phylum is that they produce sac-like asci, which is what the spores are in. So if you can think of this as being kind of like a pea pod, has peas in it, or green beans have the seeds in it. This is what an asci is. It's going to have spores inside of it. And so let's take a look. Representatives of the phylum are these cup fungi, and there's these morels, if you know these, and then truffles. So there's a lot of different species, very diverse. So mycologists, people who are studying fungi, have discovered over 60,000 species of these. They're rather complex. And so this sort of fruiting part right here, the reproductive part, this is what I meant by asci. So they're these structures that contain spores inside of them. Okay? And the whole structure that is surrounding each individual asci is called an asciocarp, the fruiting body. Okay? And now these can grow, obviously, in nature, but we can also grow them on petri dishes if we give them some nutrient agar. For example, they'll happily grow there as well and absorb their nutrients from the agar using their mycelium as that extends into the agar food source. So here is like maybe subterranean, if you will, or at least in the agar going across this way. This is where they're gathering up their nutrients down in here. This happens to be a cup fungi. Here's the asciocarp. Inside the asciocarp are these ascae, okay, and this is, or asci for plural. This is where the fungi is producing its spores. And that's what these little guys are right here. Now, it's somewhat interesting on this is that when it comes to spore, one of the traits that I'd like to discuss in this video is the coloration of the spore. the wild type is black, and then there's this mutant strain that produces tan color spores. Okay, so you can inoculate them in a petri dish and grow them containing agar. And at first, the mycelium sort of grows, and then it ultimately produces the fruiting body called the asciocarp. And so, what's interesting about this is that you can have this black strain, and it forms this mycelium, and then the tan can grow, and then these things actually can come together and unite. And so you can actually get like sexual reproduction occurring in the fungi, and it produces a heterozygous fungi. Okay, that's just what I want to get at. Okay, and so this is a good picture of that. So here you have an agar plate, and then these little black spots are the fruiting structures that you can actually visibly see, but better under the microscope. And so at first they grow, and then they start to entwine and then sexually reproduce. So the tan spores, or you can inoculate it on one half of the plate, and then the black on the other, and where they come together and where they meet is where they fuse and where they mate sexually. And so you could see this better under the microscope. You could see all these hyphae, and then these dark structures right there are the fruiting bodies. And so the fruiting body, here's a close-up, is parathecium. And inside the parathecium, these little black spots are the asci that have the spores inside of them. And so let's take a closer look at that. If you were to take the parathecium literally and scrape it out of the petri dish, let me come back here, and you like scoop this up with like a little knife, and you put it on a slide, and you're preparing a wet mount. You might put a little drop of water, you might put a cover slip on it, and then crush it with a pencil eraser. And what's going to happen, it's going to fracture open like this, and you're going to be able to see these asci right here, or plural asci. And inside, these are the spores. So these little tiny circular structures are the spores. You might notice that some of them are black and some of them are sort of lighter color, and this picture is black and white. These are the tan ones right here. These are the tan right there, and then this is the black ones over here. We label that. And so this is where our conversation is going. So here's a parathecium that's been crushed open under the microscope, and you can see the asci here. Now this isn't a great one because it's hard to see what you're dealing with in terms of the spores, but this is what I wanted to show you, that you could rupture a mature parathecium and then several asci will come out. Okay, so let's talk a little bit about the genetics on this. And so the union between the black and the tan results in a heterozygous. So as you can see, here's tan and here's black. So when this zygote is about to divide, obviously the DNA replicates, and so you can see here now it's duplicated chromosomes. So this is a homologous pair right here. And so what's happening is that when spores are produced, meiosis must occur. Okay, so we're dealing with meiosis here. Meiosis. Okay, and so let's look at this. So meiosis, if you're familiar with that, is the process of, to simplifying it, the separation of homologous chromosomes. So you have a tan one going over this way and a black one going over to this way. And then meiosis 2 is just like mitosis. So it's separation of sister chromatids. So you would get 4 spores, 2 tan, 2 black. But what's interesting about that, to increase the numbers of spores, the spores can simply divide through mitosis to produce 4 more. So instead of having just your 4 spores, you would then have 8. This would produce these 2, and then these 2, and then this black one would produce this 2 and this 2. So you have a combination of meiosis 1, the separation of homologous chromosomes, meiosis 2, which is simply mitosis. Okay, so this is creation of the spores. And then mitosis, which is down here, which would increase the number within the asci. So inside this, you'd have 8 spores. Okay. Now, You might be familiar with the crossing over event. When homologous chromosomes come together during prophase, sometimes there's a physical exchange between non-sister chromosomes, non-sister chromatids. And when there is a physical crossing over, the chiasmata transfers like a little bit of black over on this one and a little bit of tan over on this one. And so when meiosis 1 occurs after crossing over, you're going to get two cells which are mostly tan but a little bit of black, mostly black with a little bit of tan. And so after meiosis 2, do you notice here there's 4 spores, but one's pure tan and this one's black. This one's going to be tan and this one's going to be black. So curiously, you get 4 different spores versus getting 2 different spores. Okay, so my crossing over increases variation. So this is the point I want to emphasize. This recombination event that occurs between homologous chromosomes results in a tan. So that one right there didn't cross over. That's a tan. This one did not cross over here, which is a black over here. So this one is non-crossing over. And then these are the recombinant ones, there and there. So when you see under the microscope an ASCII that has sort of an unusual pattern of spores, now remember mitosis increases the spores to 8. When you see odd patterns like 2, 2, 2, 2, and let me continue with this, or a pattern of 2 tans, 4 blacks, and 2 tans, that's an indication that crossing over has occurred. Non-crossing over would be, let's see, look at this one right here, you can see it really well, would be 4 tans and 4 black, or 4 black and 4 tan. But whenever you see weird things like 2, 2, 2, 2, or in this case 2, 4, 2, it's an example of crossing over. So this is kind of neat. This is like, for example, if you're familiar with fruit flies, you could just simply look at the offspring and know whether or not the offspring are showing crossing over or not crossing over. In other words, like the parent or recombinant, unlike the parent. And so when you have patterns of 4, 4, that's no crossing over. So 4, 4, no crossing over. And when you have unusual ones like this means that there's crossing over. This is 4 black, 4 tan, 4 black, 4 tan, or tan black, sorry. And then these are the unusual ones. So let me draw your attention back to a previous slide here. Do you notice how when there's 4 tan, 4 black, it's not crossing over. And then when you get these unusuals, it's because there's recombinants. Okay? And so these are the recombinant ones that occur during meiosis. And so when you look at slides like this, you can literally see this is a little easier for us to see. The 2, 4, 2 is recombinant, or the 4, 4 is non-recombinant. And again, 2, 2, 2, 2 is recombinant. 2, 2, 2, 2 is recombinant. And you're like, well, what's the point of this? Well, these are the offspring. And if you were to count the total number of offspring and then divide, or look at the number, if you look at the number of recombinant over the total, it'll tell you how frequent recombination occurs. So let me repeat that. So if you look at the number of recombinant over the total, it'll tell you the frequency, the percentage multiplied by 100, the percentage of recombination. And it's like, okay, so you go about your business and you're like, okay, this is a non-crossing over, this is a crossing over, this is a crossing over here. Again, you just keep counting and counting. And because the frequency of recombination occurs as a measure of how often crossing over occurs, these things are connected. We could say that, for example, if we're counting, like, for example, 30% of the spores are showing crossing over, we say to ourselves, you know, the truth is, it's going to be half of that, or 15%. And it's like, well, why is half of that? Let me come back to this. So back over here, why is it half? Because when you look at this slide, even though there's 8 spores in here, only 4 of them, or half of them, were the result of crossing over. So when you figure out the total number of recombinants, getting back over here, like say that there was 30, you really have to divide that by 2 in order to get the real answer, which would be 15% recombination. And so what does that tell you? Well, since we're only looking at one trait, let's just pull it right there and say, well, this is the gene for color of spores in the Sordaria. If there was a second trait involved, if we were looking at 2 traits, we can actually determine a map distance with this. But let's just say it's from the centromere. So we could say that that distance from the centromere is 15 map units away. And so we can determine a gene map by looking at frequencies of recombination in Sordaria. So it's kind of neat. And so I hope you enjoyed this video on meiosis in Sordaria and how that relates to recombinants in the spores, and which leads us to be able to map the location of the gene that produces coloration in spores in Sordaria. Thanks for watching.